Good morning, and thank you for joining us all today. My name is Stephanie Bolton, and I help lead viticulture research and outreach for the Lodi growers, along with the Lodi Winegrape Commission's research committee. 1 year ago today, I stood up here and told you that I have viruses. This is still true, but since then, I have been tested. It's leafroll 3. We have a really great lineup of speakers for you this morning. Dr. Kent Daane will teach us about vine mealybugs. Dr. Mark Fuchs from Cornell will talk about viruses. Professor Jerry Peterson flew for 38 hours to get here from South Africa to help us with leafroll virus management. And Dr. James Stamp will teach us about planting material. You get to not only learn something new, but also listen to some cool accents. Each of these speakers was carefully selected because they really care about helping us, they really know what they're talking about, and they are great at communicating to a grower audience. If at any time during their presentations you have questions, please write those questions down on the index cards that are in the center of your table. We'll collect those index cards just before our question and answer panel later in the program. In addition to learning through presentations from our wonderful speakers, you'll have time to interact with the many businesses and organizations which are important to reducing viruses in California. We have associates in insectary and parabug in that back corner. They work with mealybug biocontrol insects. We have 2 pheromone mating disruption companies. Sutera will be passing out again this year, thank you so much, free mealybug traps over there. We have every single commercial virus testing lab in California in that back corner. We have BASF, who has a new ant control product they want to talk to you about. VineView is here, and they do leafroll virus aerial imaging. And then we have 8 CDFA registered nurseries over here. We have Foundation Plant Services, National Clean Plant Network, the USDA Tree Assistance Program, and the San Joaquin County Ag Commissioner's Office, all represented today. So I think you can tell it's very important to our industry that we get a handle on this problem. So why are we here today? We're all super busy. We all took time out of our demanding schedules to make today happen. It's not quite raining yet, so that's not the excuse. We showed up. Here's why we're here. It's simple. We've got a lot of these. Grapes, vineyards, handsome guys named Aaron. We've got a lot of these. Vine mealybugs. Too many, really. Almost all of us have started to see more and more of these red leaves. And all this is costing us a lot of these. So what we did is we formed some teams to try and figure out what to do in Lodi about mealybugs and viruses. The first team we formed was the Mealybug Biocontrol Research Focus Group. It was important for us to have a mix of growers, PCAs, extension, and scientists. This successful group hosted a Mealybug ID field day in August 2017, and they also created the beneficial insect risk chart. Next, we formed the Lodi Grapevine Virus Research Focus Group, also a team of growers, pest control advisors, scientists, viticulturists, and extension personnel, and nurseries. And we're lucky enough to have Professor Peterson and Dr. Mark Fuchs as our experts willing to give us advice and assistance as we navigate our local virus situation. It helps that many members of our team have a financially vested interest in finding practical virus management solutions, and that they are willing to work towards a greater good for everyone. After all, our Lodi Winegrape Commission motto is, "A rising tide lifts all boats." We meet on the first Wednesday of every month at the Velvet Grill Diner in Lodi to discuss viruses for hours over pancakes and coffee. This photo is from yesterday's meeting. We started learning everything we could about virus management from anyone who would talk to us. We were in shock for a while at how bad the situation was. Then we accepted the reality that in California we have prolific, widespread populations of vine mealybugs, and that there is a significant amount of virus inoculum or source points in our vineyards. We also realized that no one locally had a good working virus management protocol. We became determined to find solutions to help ourselves and our fellow growers. It became clear early on what needed to happen. So we wrote a grant proposal for an integrated outreach project focused on grapevine viruses. Luckily, we were funded by the American Vineyard Foundation and the CDFA PD Board. If you are a reviewer or a board member, thank you. This project includes virus testing and the collection of case studies, the writing of virus management booklets as information becomes available, monthly meetings for growers and for the focus group, the formation of demonstration vineyards, and lots of outreach. We've also been successful in bridging the gap between growers and different sectors of the industry as we work together to find virus management solutions. We believe that when we all learn together, we can better work together and we can find solutions faster. Consider yourself involved. Our main strategy right now is to decrease vector populations and to lower virus inoculum. In other words, we need to lower the spread and numbers of vine mealybugs and rogue virus-infected vines out of our vineyards as that is financially feasible. It's going to take a lot of teamwork and help from the growers, nurseries, FPS, the CDFA, academia, virus testing labs, wineries, extension, IPM companies, pest control advisors, and viticulturists, county ag commissioners, and regional associations. Basically, if you're in the room today, we need you. We need to stop pointing fingers and realize that we all contribute to the problem and to the solution, and then decide what action each of us can take so that we can all move forward together. Step 1 is education. Last night, we hosted an intimate grapevine virus influencer dinner where about 20 people talked through their long-term strategy ideas for grapevine virus management in California. Later today, we'll host meetings for nurseries, virus testing labs, and extension, each with growers as part of the conversation. We're definitely making progress in the area of communication between industry sectors. Many of you have been joining us for our virus outreach events. Be sure to give your email address to Julie or Shirley at the sign-in desk if you want to join our new Grapevine Virus email listserv so that you can stay informed. Our virus focus group plans today's indoor meeting for the spring each year, and we also plan a hands-on meeting in the vines each fall so that you can learn how to scout for virus symptoms in red varieties. Last fall, we also learned about mealybug biocontrol, virus testing, and the three-cornered alfalfa hopper. We're realizing that we need to focus our management efforts on these guys: leafroll 3 virus, vine mealybugs, and ants. Does anyone recognize this photo taken by Mario Salinas, who's in the house today, by the way? It was on the cover of the recent PD GWSS newsletter, and it will hopefully convince more California growers and pest control manufacturers of the need to control ants in vineyards. This little ant is carrying a mouthful of mealybug eggs along a drip irrigation line, off to start a new colony of mealybugs on a grapevine. We've been collecting case studies to investigate the potential role of leafroll virus in a mystery vine collapse where large patches of vineyards are dying all over Lodi and in other parts of California. Vines push out fruit and appear healthy, and then all of a sudden, in a matter of weeks, the entire vine is dead, dried out, and even splitting along the trunk in some cases. At this point, it is looking like a combination of freedom rootstock and maybe others, leafroll 3 virus, and a third stress, which could be trunk pathogens, vitiviruses, crop or water stress, etc. Over time, these patches become larger until the vineyard is no longer profitable and needs to be removed. We're creating a grapevine virus workbook for growers, starting with small draft booklets that can be handed out before the final version is ready. This way we can get some tips to our community now, and we also get a chance for growers and others to help us improve the content. We're sharing hard copies of these booklets with anyone who wants one, and the goal is for other wine growing regions in California to customize them for their growers. The first booklet we made is called Nursery Ordering 101 Viruses, and it teaches growers how to order CDFA certified material, which is actually quite confusing, and then how they can take some responsibility for starting with cleaner material, which includes asking for it, extra testing in the beginning, and good record keeping. If you're planning on ordering plant material soon, you should definitely pick up a copy back there. Our second booklet is on vine mealybugs and is available for the first time today, hot off the press. By taking a draft copy, you agree to provide comments and input. We have 50 copies here with us today, and they come with a new custom Vine Mealybug flagging tape. Thank you so much for all you're doing to lower the amount and spread of vine mealybugs, to decrease virus inoculum in the vineyards you manage, and to educate yourself. We are grateful to the CDFA's PD Board and the American Vineyard Foundation for funding, and UC Cooperative Extension for sponsoring our room today, and to the members of our Lodi Mealybug and Virus Research Focus Groups, steered by the Lodi Winegrape Commission's Research Committee, for all of their dedication to helping their fellow California growers.
Working together in the wine grape industry has been really exciting to see over the last 5 to 7 years, watching wineries and vineyard management agencies that are all competing in the same space, all really group together to solve this problem. I think that's been the most exciting thing to see this morning.
Next up, we have Dr. Kent Daane, the smartest mealybug guy we know. If you own a Lodi vineyard or if your paycheck depends upon Lodi grapes in some way, you should definitely tell Kent thank you today. He released populations of anagyrus wasps in Lodi years ago, which have played a really significant role in parasitizing vine mealybugs here. Let's all give a warm welcome to Kent.
I'm going to give an overview of vine mealybug as a vector of leafroll. We're not going to talk about red blotch here today, the other red leaf disease, and the different types of control programs we've got. This is going to be more of a general presentation, but I'm going to stick around for the work groups afterwards so we can get down into the weeds about the nitty-gritty details of all of these different control tactics. And I want to thank all the co-authors, friends that I've worked with, colleagues in the lab, and leaders of other laboratories that have helped with this project. So vine mealybug is one of a number of invasive species. It's one of 4 invasive mealybugs. We've got the vine mealybug, the obscure mealybug, the long-tail mealybug. Even the Gills mealybug is not from California. The only California area mealybug is the grape mealybug. It was the #1 vector of leaf roll before the vine mealybug arrived. Vine mealybug is much different than these other mealybugs in terms of its effectiveness at moving the leaf roll around, particularly because it's got so many generations per year. And the crawler, the smallest stage, is the best stage in terms of efficiency in moving it. Since it's got probably 6 to 7 generations per year here in Lodi, you've got a lot more crawlers, a lot more chance for the vine mealybug to move the pathogen around. So basic outline, we'll look at mealybugs as invasive species, their damage, not just direct, but how they work in terms of being a vector of leaf roll, biological controls, mating disruption, chemical controls. Then I'll try to bring this all together to talk about practical applications and conclusions. So as I mentioned, vine mealybug is one of these invasive pests. The next one is going to be the spotted lanternfly, which is now on the East Coast, can be devastating of grapes. Hopefully the Rockies, the mountains, the deserts keep it from getting over here. Vine mealybug, like the obscure mealybug, came from someplace else. This is the obscure mealybug, 3 generations per year. We find it in Lodi, although its numbers now are harder to find because of all the applications for vine mealybug. It came from South America. It spread throughout the world, mostly on nursery plants. When you go into any of the shopping malls and you see things like ficus or on the umbrella leafy plants, and you see mealybugs on it, it's commonly the long-tail mealybug or the obscure mealybug. So, spread throughout North America. Interestingly, in South America, they don't have as many problems with the Argentine ant as we do up here. That's because the Argentine ant in South America keeps its own numbers down fairly low. One colony of Argentine ant recognizes another colony just down the street as an enemy, and they kill each other. Here, we've got one large sister population of Argentine ants that all work together. That's why it became such a pest here. Vine mealybug, in particular, came from the Middle East, Mediterranean region. It's now a pest in South Africa, it's a pest in Chile, it's a pest in Argentina, it's a pest in Peru. If you go to France, they don't consider this to be as much of a problem as we do here. That's because it's probably made up of a bunch of sister species. And in fact, when we got into the weeds on this, looking at the molecular work, we can see the California and Mexican population. This is kind of a phylogeny tree looking at the relationships of different populations of vine mealybug. California and Mexico have got a very, very similar population. So you would assume it came from the same spot. When we started working with this, we thought it came up from Mexico. I went to Mexico to work with colleagues there. They thought it came down from California. Well, in fact, we know which farmer brought it in. He farms in Coachella. He farms in Hermosillo, the two spots it started. We know the first find of vine mealybug was in his next door neighbor's spot, and he had bragged that he had gone to Israel and picked up some wood with table grape material that was better suited to the desert region. And in fact, when we look at material from Egypt and Israel, we can see it was very, very similar to a population from Israel. And we see how far apart this group of Israeli-Egyptian vine mealybug is from populations found in Europe. So that's one reason why it might be a little bit harder to control here. It might be a little bit better suited to hot climate. We can see for Gerhardt, the South African population probably came from Europe, probably from Spain or Portugal. Argentinian population probably came from Spain. And we've got two other groups here from Turkey and Tunisia. So each of these different populations will have different biological characteristics, making it easier or harder to control depending on how many generations they have, how much fecundity they have, meaning how many eggs they've got. Once in California, we found it in Coachella. Walt Bentley tells the story of looking at those 20, 30 vines that were infested, discussing whether or not they should pull the entire vineyard out or just nuke it. The decision was, let's just put on a lot of lanate and lorsban and it will probably kill it all. It then made it into Southern California, around Bakersfield, around Fresno area. We now suspect these were infested table grape clusters that could not be marketed, so they were sent into the Central Valley to be crushed for juice grapes. We now know that the vine mealybug, a small percentage, can actually survive the crush going through wine grapes, and people would take that crush and spread that pumice out on the dirt roads to lower dust. So a tiny percentage can survive, so we probably got, because the first finds were around juice-producing plants, we probably brought it up from Coachella, spread it on the roads, and it made it into the vineyards. And then, of course, there was explosion later in the early 2000s because it did make it into the nursery material. The nurseries were doing what they were supposed to do to control for exotic pests, because everyone at this time was concerned about Pierce's disease and glassy wing sharpshooter. And the control for that was to spray the nursery material with Pravado, imidacloprid as a foliar form. That's great for glassy wing sharpshooter. We know that doesn't work for vine mealybug. You've got to use Admire or one of the other systemic neonicotinoids, or Belay, a foliar neonic. So what happened was that it got into the nursery material, it got down on the roots, and we started planting vine mealybug throughout California. I mentioned it's a lot worse than the other invasive species. I take this because of 4 different reasons. First, it causes more damage just in its feeding because it's got more generations per year. Grape mealybug, about 2 generations per year. Obscure mealybug, about 3 generations per year. Vine mealybug in this region, about 6 to 7. It can have as many as 8 generations per year down in Bakersfield. It's going through a generation right now. Grape mealybug has a diapause. It stops sometime in September, October developing, just stays as an ovisac or a first instar. Vine mealybug in January, if you kind of get a nice warm day, it'll start to feed, it'll start to develop. We oftentimes don't see this because we think that nothing is happening underneath the bark. But we're going through a generation underneath the bark in January and February, and so we get these off-sync populations. Right now, I can go to any of your infested vineyards, and I'm sure I can find crawlers, second instars, third instars, and even some adults. And that makes some of our chemicals less efficient, especially for the organic growers, because those materials really target the first instars. It also feeds on the grape leaves far more prevalently than the grape mealybug. That allows it to get up above the cluster. It allows it to rain honeydew down on the cluster, causing more damage. Much more important for table grape growers than when we consider it as a pest, just because of its ability to vector. So where we've got typically, the grape mealybug would have populations just like this. The vine mealybug can really explode in densities. But what we're interested in is its issue in terms of being a vector. Classic picture from Deborah Galino, close-up of a leaf infected. Here's some of the work I did with Rodrigo Almeida's lab. First bit of information you have to remember as any of these mealybugs, as being vectors of leafroll, is they can acquire the virus very quickly. So within 1 hour or less, they can pick up the pathogen. It goes up to about 24 hours. That's what that graph showed, and it kind of maxes out. They can inoculate the pathogen very quickly. Within about an hour, it can move it, once feeding on the vine, into a clean vine. So an hour acquisition, an hour inoculation. That means for me that if you're using one of these great materials, any of the systemic materials, Movento is a systemic material. The neonics are typically systemic materials. The mealybug has to feed on the vine in order to die. With Movento, it has to feed for quite some time to die. So we always say wait 3 weeks, wait 4 weeks sometimes, depending on where that mealybug is. So remember, acquisition and inoculation within 1 hour, and we've moved away from the lanates. We've moved away from the toxic contact materials to systemic materials, so you can have mealybugs brought in by birds, by equipment, by air. The crawlers you're not going to find anyway because they're so small. They can feed, they can transmit the pathogen, and maybe 3 or 4 days later they die, but as a vector it's done its job. One of the other things I like to point out is that so many vector-pathogen relationships, when we've got an insect and a pathogen, it's a long evolved situation. Not so with these mealybugs with leafroll. This chart just shows the relationship on the left of different leafroll viruses, and when this was done, they were going up to 8 or 9 or something like that. And on the right, these are just species names of different mealybugs, so I'll give some pictures of the 4 we're talking about. And we see all the arrows going basically to leafroll 3, because that's the most common one, so it's the most tested one. But the bottom line is that there's no real evolved relationship. Almost any of these mealybugs can be a vector. So right now, if you're in El Dorado County, you're worried about the Gills mealybug. This was first found in California on pistachios, then almonds. Now it's on grapes in the foothills. We tested it. Yes, it also is a vector of leafroll. So there's no evolved relationship. Any mealybug should be considered bad. Another point that I like to pull out, we tested these different species, and we can see here in the laboratory they're between 5 and 50% efficient. In the field, between about 1 and 30% efficient. Well, what that means is these are very effective vectors. Glassy-winged sharpshooter has to feed and probe and feed again and probe again before it transmits Pierce's disease. We're taking one crawler, something you can barely see, and putting it on a vine, and sometimes as much as 50% of the time, that one crawler is transmitting that pathogen. So it's a very efficient vector, which makes control a little bit more difficult, because it brings the number of acceptable mealybugs down to such a low level, I don't think we can attain that with insecticides, biocontrol, mating disruption. I think we have to focus on roguing, because we're never going to get the mealybugs down to a level that's acceptable for them to control as vectors. There are some differences in efficiency between mealybug species. We're looking here at young mealybugs, old mealybugs, and we're looking at, on the top, vine mealybug versus the other invasive species, obscure mealybug. And if you look at the inoculation, the graph on the right, it just shows that the vine mealybug was a more effective vector than the obscure mealybug. I've been trying to figure out why that might be, why the crawlers might be more efficient than the later stages. I think maybe it's because the vine mealybug, because it has so many generations per year, it feeds more. If you go from a baby to 6'5" in 4 years, you're feeding a lot more than if you're doing that in 30 years on a day-to-day basis. So maybe by feeding more, they're acquiring more of the pathogen. I think the crawlers might be more efficient because they might just be sloppier. They've got this thing in their esophagus, this pump to bring the fluid in, goes to the digestive system and comes out as honeydew. And maybe as an adult, that pump's a little bit more muscular, a little bit better at bringing the material up and not being sloppy, like a kid with a straw blowing bubbles back into the milk, something like that. These details we haven't worked out, not that important. What is more important is that they do lose the pathogen with every single molt. So if it feeds on an infected vine, it's not going to pass that pathogen on. If it goes to a clean vine, molts, it's not going to pass it on to its offspring. Some vectors can do this. It makes control much, much harder. So once it's fed on an infected vine and acquires the pathogen, 3 or 4 days later, it loses that virus within itself. One of the other aspects of this, and I'm going to run out of time, so I'm going to start speeding this up. One of the other aspects that we always get questions on is how soon do we see symptoms after the mealybugs feed? This becomes very important for new plantings. Did I get it from the nursery? Do I have mealybugs in the ground that are feeding on the roots, coming up, and now I've got the pathogen? So what Kai did with Rodrigo and myself is that she went out and we inoculated 10 mealybug crawlers per plant on July 19th. And we can see the arrow following that, the little circles at the very bottom of the graph. We saw no symptoms that year. So a July inoculation, no disease symptoms the rest of that year. The next year, we were starting to see symptoms around June, and the following 3 years, around mid-May, we started to see symptoms. So if you are inoculated this year, in the middle of the summer, you're probably not going to see symptoms until next year, and then you'll start seeing them around May or June, every single year thereafter. So, what about control? I'm not going to go into detail on any of these subjects except for chemical control. We'll start off with natural enemies. There are a number of good predators out there. Many of these you can purchase. We've got associate insectaries here. Brett's an expert of all of these. He can tell you the pros and cons. So I'll just give you a generalization. We've got the going around the clock. We've got the green lacewing. There's also the brown lacewing. You can't purchase that. It's typically more common in the spring than in the summer than the green lacewing. We've got the mealybug destroyer. It's got the larva that's got the dreadlocks on it where it looks like a mealybug, so it works a little bit better in the presence of ants. Years before, I've shown videos of all these natural enemies, so we're just going to talk about their basic biologies here. The mealybug destroyer works better in the presence of ants, but it tends to leave if the mealybug densities get a little bit low. There's other kinds of predaceous ladybird beetles that are native to California that also have got larvae that have the kind of dreadlock look to them as well. Sometimes we release the mealybug destroyer, we see these others. All of their issue is overwintering and the density of the mealybug. The little orange larva is a sesodemyid midge. These are common in vineyards with large numbers. And in fact, that's what I think is one of the issues with most of these predaceous natural enemies of mealybugs, is that to stay in the system, they like to have a vineyard with a lot of mealybugs in it, because that's where they're searching to put their eggs. Remember, these tiny little first instar mealybug destroyers can only crawl around on a single vine. They cannot go 20 vines down or across the road or across 5 rows. They just aren't that big. They're not that good of dispersers. So the adult female, mom, will fly away and try to find a vine with a lot of mealybugs on it. They're not lost to the system in Lodi, but they might end up in your neighbor's vineyard if he or she has more mealybugs. That's why area-wide controls are oftentimes important. I really like, at low densities, the parasites a little bit better. This is Anagyrus pseudococci. It was first imported in the 1920s, the 1940s for the citrus mealybug, the Comstock mealybug. We brought material in from Spain, which I like a little bit better. It probably is a different species. We're not separating these out yet, but it attacks the smaller stages of the mealybug. That's why I think it's a little bit better at controlling mealybugs throughout the year. There's also this Coccidoxinoides peregrinus. We brought this in with von Walton from Stellenbosch. It has a bigger role, I think, in South Africa than it does here. We're finding it here, it's established here, but we don't see it in the kind of numbers as we see Anagyrus, in part because right before it gets to a pupa, the mealybug kind of senses something's going wrong, and it drops from the vine or it goes in the cracks and crevices on the bark, and we just don't see the pupa on the leaves. These parasites do have problems. All these control tools will never get you down to zero. We can't eradicate mealybugs, and I'm going to stress that again. So for example, Anagyrus is the best of the parasitoids that I think we've got. One of its issues is that it starts too low in the season. We see here in May, parasitism is less than 10%. That's because it's got this overwintering biology where it doesn't want to come out from the mealybug as an adult until the mealybugs are out on the vine. It doesn't search well for mealybugs underneath the bark. So it just evolved not to be present, not to be searching until the mealybugs are starting to go out and onto the leaves. That's good for it, that's bad for us. It reaches high levels of parasitism late in the season, but remember, as a vector, as an efficient vector, we're really going for zero. So it helps, but it's never going to drive you down to zero. There's always going to be mealybugs located underneath the bark that this natural enemy doesn't get to. And so ants are also a problem for almost all of these predators, especially the parasitoids. We've shown data before, if you've got ants, you've got less parasitism. What I'm showing here are two types of controls. We worked with the liquid baits in tanks. Typically, you can purchase these things. They've got commercial products. We should have gotten a Section 18 for boric acid. I still want to do that. Eventually, I will get to it. But it became labor intensive. We've also got these gels that are being tested. These also have to be broadcast spread. This also became labor intensive. So all of these ant controls cost a little bit more money than they work, but they cost a little bit more, they take a little bit more time. So that's adding to your mealybug controls. We worked with Jocelyn Miller, a really great pheromone chemist out of UC Riverside. He developed pheromones and synthesized pheromones, identified and synthesized all the pheromones for all the mealybugs in California. And the vine mealybug was so cheap compared to the great mealybug to produce. Initially, we were just trying to do this as a monitoring tool. And we did this before vine mealybug was even up here in Lodi. And Walt Bentley and I would go around and give talks saying, hey, use these traps. These are amazing. Use these traps because the mealybug's going to be here one day. And when you find it, tell everybody, and we can go in and we can spray it. Well, you guys were not so vocal when you found it. So it got up here, again, moved on the nursery, and all of a sudden it was in Napa, Sonoma, and eradication was just not even an issue. Napa tried, and they couldn't control it. So we found that not only was this good for monitoring, but it was so cheap to produce. The base of this formula is the same base in the perfume Charlie. And so what happened was that there were groups, a company in Japan that was mass producing these perfumes, and it was a simple one more step to mass produce this pheromone, that synthesize the pheromone. Still, it's expensive, and we'll talk about that later. We initially tested this with Sutera products. Initially, it was a sprayable formulation. We were almost always getting a reduction by at least 50%. The lower the mealybug, the better it would work. We almost always had a trap reduction, meaning we shut down the pheromone traps, which was good, and we typically got a reduction in crop damage, which is good. And this particular trial went from about 9% to about 3%, so about a 60% reduction. We then tested this as a pheromone in a lure because Suterra initially was going to market this to the organic growers. The encapsulated formulation at that time, they're working on getting it registered for organic growers, but at that time it was not going to be registered for organic growers, so they came up with the dispensers. I'll show you data next with Pacific Biocontrol. It's great to have another player in the market because maybe price competition will lower the price for this a little bit. So Suterra product on the left, Pacific Biocontrol on the right. I'll use same kind of data for both groups. I'll use last year's data from Pacific Biocontrol. We can see here excellent trap shutdown. What I really liked about their isomate dispenser is it clearly lasted the entire season, so we're putting it out next week to put it out really early this year and see if we're still getting trap shutdown in October, November. Here's one I want to point out to get us back on the idea of area-wide control. This is the trial of Pacific Biocontrol. Blue is no mealybugs, red is a lot of mealybugs. We've got yellow was the control, red was the pheromone. The chart down below was wind direction coming from the northeast. This is before we put pheromones out. This is after we put pheromones out. You can see the flip-flop. But the most important thing to see here is that on the windward upside, in the control, a lot of mealybugs, we're getting control even in our control plots because the wind is blowing the pheromone. But on the north side, we're still not getting as good of control because the wind is blowing the pheromone away. If you've got 2 acres, if you've got 5 acres, and you think, oh, in that 2 acres, I'm going to put out mating disruption to knock this back, it's not going to work in those 2 acres. The larger area you use with mating disruption, the more effective it's going to be. So I'd like to see area-wide control using mating disruption and really target the insecticides where you've got bad populations to try to bring that down. So I'm going to finish this off with insecticides. We initially were working on something other than Lorsban because we lost Lorsban. It was actually an in-season product at one point when I started. Then we lost it as an in-season. Walt Bentley got a Section 18 as a delayed dormant product. So then we were looking at something that would be as good as Lanate. We've now lost Lanate. And we came up with Applaud. We've now lost Applaud. And we came up with the neonicotinoids through the drip, which we still have and I think still work very well. Later on, along came Movento. Belay is another good product. There's others, but we really liked this combination of a neonicotinoid and the Movento Spirotetramat because we like to go one product than the other. If you're going to work on resistance management, work with your neighbor because you've got their mealybugs and vice versa. We had to find out how Movento worked. Remember that it's sprayed. Let's see if this will start to go. It's sprayed as an AI. It goes into the leaf and it gets metabolized to Spirotetramat enol. It's the enol that kills the mealybug, not the AI. So the first thing it has to do is get changed in order to work. So it goes through this different string of chemicals, of metabolites. The enol, the one that kills the mealybug, is the first one in the chain. Once it goes beyond that, you've lost its ability. So you want it to convert immediately to the enol and then stay as the enol as long as possible. We use this machine called an HPLC to look at the amounts of Spirotetramat and enol in vines throughout California to figure out why some growers were saying Movento did not work this year. Because again, it's the best product that I can think of. Here's one example. This is 5 days after treatment. These are 20 different vines. In red will always be the enol. In white bar will always be the Spirotetramat. This just shows in this particular vineyard we got 100% conversion to the enol. That's good. That's what we expect. We also see that there was about a 10-fold difference in the amount of the enol in leaves from different vines. So different vines are going to have different rates depending on how they acquire the material, how the spray goes out. Maybe it was our fault where we collected the leaves. Maybe some of the leaves inside didn't have as much of the product as those outside. 72 days later, we still see this difference with some vines having more than others, but we also see a lot of the product still in the vines. So what are some of the quirks with using Spirotetramat, using Movento? In this particular vineyard, 5 hours after application, we've got mostly Spirotetramat. So we know it does take some time to do the conversion. 4 days later, we've got 50% spirotetramat and 50% enol. For some reason in this vineyard, there wasn't 100% conversion. The spirotetramat won't kill the mealybug, so half your product was lost in terms of its effectiveness. Maybe it has to do with the stoma being open or closed. Maybe it had to do with the surfactant that the grower was using. Maybe it had to do with vine physiology. These are all questions we're trying to work out. One of the other things to note, 184 days after application, that spirotetramat really still had not converted over. So I think the conversion is immediately when it goes from the leaf into the leaf, rather than going from the trunk back up to the leaf later. I also want to point out that 1 year later, we're still finding spirotetramat enol in the vine. It's at a much lower level, but it's in the vine for about a year. That can be good. It's in there for a long time killing. That can be bad. You get down to a really low dose, and what does that lead to? It leads to resistant possibilities. What about movement from the leaves? These were all data from the leaves. Everyone wants to know, what about the trunk? That's where it's hard to kill the mealybugs. It's really hard to kill mealybugs on the trunk. I haven't found a good way to do it yet. We spent a year working with Movento in different ways of applying it. But when we do find it on the trunk, when we do find it on the roots, it's not that low in parts per billion or parts per million. What we find instead is that it's spotty. So when we spray it on a vine, we collect the leaves, we can spray it on one half of the vine, and 10 days later we find it on leaves on the other half of the vine. We spray it in shady leaves, it's on sunny leaves later. So it spreads evenly throughout the leaf. We spray it on the leaves and we take trunk samples, and half of the samples have 0 spirotetramat enol and half have a lot. So it's spotty, and it's even spottier going down to the roots. We don't know why yet. I'm asked all the time about dose, about rate. We want 8 ounces, 12 ounces, 24 ounces. So we're going way above label rate, and we're looking at 1, 7, 30 days later, spirotetramat on the left, enol on the right. Take-home message is the spirotetramat, the active ingredient, is the way it should be. It goes up with increasing doses. The enol, again, doesn't always follow that pattern. And it has to do, again, probably with vine physiology, something that's going on in that conversion that we don't quite understand yet. I'm asked about post-harvest applications. I prefer to see a plot or something else go on if your harvest is really late. The leaves have to be actively photosynthesizing for that conversion to happen. So in this one, we sprayed really late in a radial Thompson seedless, and we have nothing but spirotetramat. We have no conversion. If you're a table grape grower in Bakersfield and you're putting this on in July after harvest, yes, you probably get conversion and it probably has a post-harvest impact. But if you're harvesting in October, late October, I would try a different product. So finally with Movento, we had a number of failed vineyards where they put on Movento. We had terrible populations of mealybugs. We went out with our machine, we looked for enol and spirotetramat. That's where the spirotetramat should be. That's where the enol should be. So far we've done this 4 times. In all 4 cases, there was an application error. So I think the application is really key with Movento. We don't know all the way it should be done, but just as kind of general rules using Movento, if you've got underneath the bark adults that are putting in ovisac, Movento won't kill those because they're not feeding. They're putting out an ovisac. If you've got eggs, they're not feeding. It won't kill those. It's not getting down to the trunk in the same dose that it's getting to the leaves. So those mealybugs on the trunk aren't going to die a week after you spray or 3 weeks after you spray. It's going to take a much, much longer to do that. There's differences in vine physiology where this product goes. So in conclusion, lots of different mealybug species in California. South Africa has the same issues. We're unique in that we've got almost everyone's mealybugs. Lots of chemical controls. I listed some of my favorites, but Movento, Belay, Admire, all the neonicotinoids, Applaud, which people are not using as much anymore, Savanto, all have knockdowns. I like probably Movento as the best, but the others work very well as well. I think Movento, Belay is what I'd like to see back and forth every single year. There are excellent biocontrols for grape mealybug, not so much for the others. Talk to Brett, talk to us later on in the breakout sessions on how to use these natural enemies. I like mating disruption, but it's got to be used as an area-wide program or larger acreage. These are all tools, nothing eradicates the mealybug, so no single control works. Again, area-wide programs are best. Your neighbor's mealybugs are your mealybugs, their viruses are your viruses. Thank you very much.
Lodi Wine Growers has been great. They do a tremendous amount for this community. Their communication is fantastic with growers in all aspects, and especially most recently they've done an excellent job of giving growers a resource to find information, especially about topics like this.